Awesome. So it looks like we have some people trickling in. Hi, everybody. My name is Alexa Tinsley, and I am the Director of Programs here at Jett Foundation. Thank you all for joining our community webinar series today. We welcome Dyne Therapeutics. We're going to get started. I'm going to give everybody about 30 seconds just to hop on. We do have quite a few people logging in this afternoon, so we'll be back in about 30 seconds. All right, it looks like the numbers are pretty steady, so I'm going to go ahead and get started. Thank you again for joining us today. Today we welcome Dyne and their team to discuss transforming the treatment paradigm for Duchenne. We are very, very grateful to our partners like Dyne for helping us make all of these webinars possible, bringing them right into your homes and helping us educate the community near and far. So thank you, Dyne, for being here. We do welcome Molly White, Vice President and Global Head of Patient Advocacy and Key Opinion Leader Engagement, and Ash Dugar, Chief Medical Affairs Officer. We're very excited to have them join us today. Before we get started, though, I do want to mention just a few things. Today's webinar will be 20 to 30 minutes, and we'll have a live Q&A at the end of the presentation. It is being recorded, so for if any reason you aren't able to stay the whole time, we will have it up on our website later for you and our YouTube later for you to view. There is, for the end, a Q&A. It looks kind of like two little chat boxes at the bottom right of your screen. That's what we will be using to answer questions at the end. So please submit your questions through there. We do our best to see if there are any questions that come in through the chat, but that is our primary portal for answering questions at the end. So with that, I will hand it off to the Dyne team and Ash, and we will start the presentation. So let me just share my screen and we will get started.
Great. Thank you, Alexa. So as Alexa said, I'm here with Molly White, our Global Head of Patient Advocacy and KOL Engagement, and I'm Ash Dugar, the Chief Medical Affairs Officer at Dyne Therapeutics. First, we'd like to thank the entire Jett Foundation team for inviting us to present at this webinar to update the Duchenne community on the progress of our program, and specifically Dyne 251, our clinical candidate designed to treat patients with Duchenne who are amenable to exon 51 skipping. As always, it is a true privilege to speak with the Duchenne community, so thank you for giving us the time today. Next slide, please. These are our forward-looking statements. Please refer to our website if you are interested in more details. Next slide, please. So I always want to start with this slide. At Dyne, it all starts with our mission. We are laser focused on delivering life-transforming therapies to patients with serious muscle diseases. I see Molly's square going around here, with Duchenne being a priority. Our mission is truly core to who we are and what we do every single day. And we're incredibly excited to inform you that we've started dosing participants in our Dyne 251 program. And our focus today is on sharing an update on our clinical trial progress and details about our clinical trial design on Dyne 251. Next slide, please. Over the last few years, we have built a robust portfolio focused on rare muscle diseases. Myotonic dystrophy type 1 and FSHD in our portfolio represent opportunities to develop first-in-class therapeutics. And in Duchenne, we're building a global franchise starting with our exon 51 program. And we're going to be following this with programs for exons 53, 45, and 44. Next slide, please. We're building Dyne to be a leading muscle disease company to deliver for families living with Duchenne and other muscle diseases. Shown here are three core strengths that help us deliver on that. First is our commitment to delivering for patients. We've initiated our clinical development program in Duchenne, as well as myotonic dystrophy, and these programs have been informed by the patient communities that we serve and scientific leaders in the field. Second, we have exceptional in-house and advisory expertise with deep knowledge of Duchenne and developing novel therapeutics, and we have a world-class scientific advisory board which includes luminaries such as Doctors Francesco Montoni and Lou Kunkel. Third, we intentionally designed our program to overcome one of the biggest limitations of current treatments, which is effectively and efficiently delivering modern oligonucleotides to muscle tissue. Dyne 251, our investigational medicine for Duchenne, is designed to utilize an exon-skipping phosphorodiamidate morpholino oligomer, or PMO, to increase dystrophin expression and significantly alter the course of Duchenne. We believe our technology, our scientific platform, can better deliver antisense oligonucleotides like PMOs to muscles, including skeletal muscle, respiratory muscles, and cardiac muscle. And our technology should allow for tunable, titratable, redosable therapies with relatively infrequent dosing. And I'll get into much more of this as the presentation progresses. Next slide, please. So I know that I don't need to spend much time at all describing Duchenne to this audience. We recognize that DMD is a degenerative condition where the lack of dystrophin protein leads to muscle wasting. Muscle fibers are replaced by fatty or fibrous tissue over time. And while symptoms are seen as young as age 2, on average, Duchenne isn't diagnosed until age 4 or 5. And most individuals with Duchenne are non-ambulant by their 20s. We have learned so much more from you, the community and advocacy leaders, about how this affects families physically and emotionally. And it's really you and your loved ones that have taught us so much. So we thank you for that. Next slide, please. So this slide depicts the role of exons in producing the dystrophin protein. And I'll just go through this very quickly because I know that this is a very informed community that understands the mistakes that occur in the dystrophin gene leading to Duchenne, as well as how exon skipping works. So I'll go through these next few slides fairly quickly. A length or section of DNA is called a gene, and each gene is made up of smaller parts called introns and exons. The introns are not shown on this slide. When the body needs to make a protein, instructions in the DNA are given to a smaller molecule called RNA. And during this process, those introns are removed and all the exons are linked together to make one long chain of instructions, which are then carried to another part of the cell by something called messenger RNA. Once there, particles in the cells called ribosomes read the instructions, and this is basically called a reading frame, shown here, to make the dystrophin protein. The mRNA illustrated here comes from the DMD gene and contains 79 exons that are linked together to form the instructions for making dystrophin. And the DMD gene is one of the largest genes in the human body. Now, mutations or errors in the gene alter the instructions for making dystrophin. Types of mutations can include deletions of an exon or multiple exons, duplications of exons, or other changes. The most common mutation is a deletion of one or more exons, as shown here. And this leads to the body not being able to produce sufficient or even any working dystrophin protein. Next slide, please. On this slide, we better illustrate this by focusing on a type of deletion mutation. You can see that we show a section of those 79 exons, here exons 50 to 57. And if an individual is missing exon 52, for example, then you can see the exons no longer fit together. And this is called an out-of-frame mutation. And since the pieces no longer fit together, the machinery that reads the exons to allow dystrophin to be produced cannot read all the way through that exon chain. And this results in the inability to produce the dystrophin protein. Next slide, please. So how can exon skipping help? Well, the goal of exon skipping is to allow the body to make dystrophin protein by skipping an exon to allow the remaining exons to fit together. So exon skipping splices a gene and restores that reading frame I was talking about, and it acts as a kind of molecular patch, if you will. So in other words, exon skipping encourages the cellular machinery to skip over an exon by masking it with an antisense oligonucleotide or an ASO, which are small pieces of synthesized DNA. In this example, you can see a type of ASO called a phosphorodiamidate morpholino oligomer, or a PMO, that can skip exon 51 to allow the exons to fit together again. This allows the production of a shortened functional form of dystrophin that can be produced by the body. Next slide, please. So to date, one of the biggest challenges to developing highly effective exon skipping treatments has been the lack of getting enough of the ASO into muscle cells, such as skeletal, respiratory, and cardiac muscle cells. And Dyne is looking to overcome that barrier to be able to effectively deliver PMO, exon skipping PMOs to those important muscles. Next slide, please. So here I'll take a few minutes to describe the FORCE platform, our technology to be able to overcome this limitation in terms of delivering antisense oligonucleotides to muscle. So the FORCE platform drives our efforts to develop and deliver muscle-targeted therapeutics. It's expressly designed to address that limitation that I just talked about, which is efficiently and reliably delivering PMOs that target the genetic basis of Duchenne to skeletal, cardiac, and smooth muscle. As you can see here, Dyne's FORCE platform consists of three parts: a proprietary antigen binding fragment, or what we call a Fab, to enable targeted delivery of the ASO to muscle. On the bottom part of the slide, you can see a clinically validated linker. And on the right-hand side, an ASO, or PMO in this case, selected to target the genetic basis of disease. I'll start by describing these three components, and then I'm going to come back to the importance of targeting the transferrin receptor 1, or the TFR1, to be able to drive targeted delivery of our investigational medicine. So on this slide, you can see a Y-shaped structure, which is a monoclonal antibody. So we chose a portion of this, our Fab, as I mentioned, because we believe it offers powerful advantages over the larger monoclonal antibodies, including its smaller size, lower overall protein load, and potential for enhanced tolerability profile. The goal is ultimately to enable targeted effective delivery of the therapeutic molecule to key muscles. Number two, we selected the Val-Cit linker for our platform based on its stability and clinically validated safety and efficacy in approved products. Because this has been used in other products, we believe this further de-risks our FORCE platform. Now, we're using the same Fab and linker across all of our muscle programs. The versatility and modularity of our scientific platform provides flexibility to deploy different types of antisense oligos that are rationally selected to match the target biology for each disease. Where dystrophin restoration is the key, as is the case for Duchenne, it's critical that we select an exon-skipping PMO that targets the specific mutation in the DMD gene. So really, our FORCE platform is designed with the goal of addressing the current limitations of delivery to muscle tissue, really focusing in on the ability to not only get into skeletal muscle, but also drive into cardiac muscle. And then also, we believe this will allow for relatively infrequent dosing. Now, I'll come back to the TFR1. So the mechanism of FORCE is different from other approaches. We're essentially using the cell's natural biology to deliver our therapeutic to muscle cells. Our Fab binds tightly to something called the TFR1 receptor. And why are we choosing the TFR1? Well, these receptors are highly expressed across the surface of muscle cells, like skeletal, respiratory, and cardiac muscle, as well as smooth muscle. So the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our therapeutic ASO to enter these muscle cells to be able to target the genetic basis of disease at the level of the nucleus. So we use this receptor-mediated delivery to drive the PMO into the muscle cell. We harness what already exists on the surface of muscle cells to deliver our therapeutic with this receptor-mediated mechanism. And by targeting muscle specifically using TFR1, we have the potential for a wide therapeutic index that should result in relatively infrequent dosing and allow for safe and chronic administration. Next slide, please. So in this next section, I'll talk about some of the work we undertook prior to entering the clinic with Dyne 251. Next slide, please. At Dyne, we have not shied away from using multiple preclinical models available to us to characterize our FORCE platform and gain a deep understanding of Dyne 251, our investigational medicine. This work has been critical to inform our approach to the clinic and ensure that we have patient safety first and foremost in mind as we put together our clinical trial plans. We've looked at non-human primates or monkeys to help us understand how much Dyne 251 gets into the body and how much exon skipping occurs. Because NHPs are not a Duchenne model, we cannot look at dystrophin production in this particular animal model. We have looked at a standard mouse model for DMD, the MDX mouse, which has a mutation in exon 23. And we've also looked at patient cells as well. And I'm just going to share a little bit of this data with you today. Next slide, please. On this slide, we show our FORCE platform in the MDX mouse, as I mentioned, a standard animal model in Duchenne. You can see that FORCE resulted in robust and durable dystrophin production across key muscles affected by Duchenne with a single dose of 30 milligrams per kilogram, and we evaluated these mice just 4 weeks later. The top half of the slide shows some of our data demonstrating robust levels of dystrophin expression across key muscles. We were able to achieve high levels of dystrophin expression, approximately 50% in the quadriceps, 90% in the diaphragm, and nearly 80% in the heart. We're really excited to see these levels of dystrophin restoration, especially in the heart and diaphragm, where current PMOs have had limitations. On the bottom half of the slide, we show dystrophin localization in the sarcolemma muscle membrane after just that single dose. This localization is important to see to provide us confidence in potentially seeing functional benefit. We see profound results, including about 70% dystrophin-positive fibers in the quadriceps and about 80% in the diaphragm and heart at 4 weeks. Next slide, please. And next, we evaluated Dyne 251 in NHPs. These studies are a key step in generating preclinical data before going into clinical trials. We measured exon skipping in the quadriceps, heart, and diaphragm. And just again, as a reminder, as I said before, in this model, we cannot look at dystrophin restoration because these are healthy animals. They're not a dystrophic animal model. The key message is that with our FORCE platform, time on therapy matters. Over a 2-month period, we observed robust exon 51 skipping across muscles. After 5 weekly 30 mg per kg doses, Dyne 251 demonstrated about 52% exon skipping in the diaphragm, about 40% in the heart, and about 20% in the quadriceps. Again, really excited to see this kind of penetration, especially in the heart and the diaphragm. We also evaluated Dyne 251 in 5 and 13-week toxicology studies in monkeys. We saw that Dyne 251 had a favorable safety profile with no dose-limiting toxicity observed after this period of time in these studies. So we're very pleased with the safety profile as we advance into the clinic. Next slide, please. Great. So in this next part, I will talk about our phase 1/2 study called DELIVER. Next slide, please. So prior to starting our Dyne 251 clinical development program, we proactively obtained expert feedback. We engaged key opinion leaders or KOLs from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. Importantly, we've gathered this feedback from across the globe. And KOLs in both the US and outside of the US, including Europe, with a great deal of experience running early and late stage clinical trials, have provided advice on our overall clinical development program, including our clinical trial design, the patient population, biomarker and functional endpoints, and key safety considerations, and have really helped us learn from natural history studies. It's clear that these experts believe dystrophin continues to be a key endpoint for this condition. Now, we've also received advice directly from patients and caregivers, as well as global advocacy leaders, such as those from Jett Foundation and other organizations. We've also gotten advice from the Duchenne Community Advisory Board, comprising of advocacy leaders from many countries around the world. They've provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. And this input has been critical in ensuring a patient-centric design and has included, but it's not limited to, key considerations for choosing a clinical trial, review of and input to our informed consent form, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. And based on this extensive feedback, we've developed the clinical study that I'll show you in just a moment in patients amenable to exon 51 skipping. And I'll just describe some of the key elements. Next slide, please. So our clinical trial for Dyne 251 is a global trial consisting of essentially 4 periods. We have a 6-week screening period, followed by a 24-week placebo-controlled multiple ascending dose period, where individuals receive either placebo or Dyne 251 administered every 4 weeks. This is followed by a 24-week open label extension period where all participants will be escalated to the highest safe dose of Dyne 251, also administered every 4 weeks. And then finally, a 96-week long-term extension period. It's important to note that following the placebo-controlled portion of the study, all patients will be able to receive active treatment. Now, we plan to evaluate the following outcomes in the DELIVER trial. As it says here, safety and tolerability and change in dystrophin levels are primary outcomes measures. We'll also be looking at muscle function relevant to both ambulatory and non-ambulatory participants. So that would include assessments to help us understand changes in both lower and upper limb function, as well as respiratory function and cardiac function. With the help of our expert advisors, we're confident that this study design is robust, will measure key outcomes, and is striving to manage the burden on families. Next slide, please. So this slide shows a select set of inclusion and exclusion criteria. Much more detail can be found on our clinicaltrials.gov page, and the link is shown at the bottom here. And you can also see what open clinical trial sites we have when you go to this website. We're enrolling patients with Duchenne who have mutations amenable to exon 51 skipping therapy, as I mentioned before. And our goal is to enroll approximately 46 male participants, ages 4 to 16, who are either ambulant or non-ambulant. We do require open biopsies of the upper arm muscle, 2 biopsies, one at baseline and one at 24 weeks. And participants should be receiving stable steroid dose for at least 3 months before the start of the study drug administration. And if someone has been on exon skipping therapies, we do require a 12-week washout. Again, please go to the ct.gov website for more details on inclusion and exclusion criteria. Next slide, please. And finally, you can see a wonderful family pictured here. We've really tried to take every opportunity to listen to the Duchenne community on their needs for next generation treatments. We're working urgently to deliver for the community, including families and healthcare providers, to bring a potentially transformative treatment forward. Thank you to the families for being generous with your thoughts, advice, and for the sacrifices you continue to make. On behalf of Molly, myself, and Dyne, I'd like to thank you all for your time and attention to listen to this presentation, and we're pleased to take any questions. Thank you.
I'm making my way back. I'm here.
Hello.
Thank you guys so much. We really appreciate that. I think that this is incredible, and we're very excited for you, and I'm sure the community is as well. So we will start taking questions. I'm going to stop sharing my screen so that I can. All right. All right, so if you do have any questions, again, there are like two little chat buttons in the bottom, for me, bottom right-hand corner that you can put your questions through. All right, it looks like we do have one. So why start with exon 51 when there are already several approved therapies for that population?
Sure. Yeah, I'm happy to start, and then if Molly wants to also add, please do. So there's a few reasons for that. So we definitely recognize that there is an approved therapy in the U.S. for exon 51 skip amenable population, which by percentage is the largest segment of the Duchenne population. Outside of the U.S., except for maybe a couple of countries like Israel, there are no exon skipping therapies approved, and certainly not for 51 skip amenable mutations. So there is a huge need to serve a global population that has no treatment available to them right now for exon 51 skipping. And then also there is a need for, we believe there's a need for an improvement in therapies where they are approved in other countries. And the other part of it too is there's also regulatory precedent. I mean, there's also a standard of care that's been established. And so it also provides a way for companies in this space to be able to say, okay, where's the bar? And we want to be well above that bar so that we can provide something that's truly transformative to patients and their families. Molly, do you want to add anything?
Nope, that's exactly it, I think.
Second question, are there plans in the future to include an arm in the trial for females with just dystrophinopathy?
Yeah, it's a really, really important question. And we've had this question in the past. So not to get too far into sort of drug development, aspects here. This is a phase 1/2 study. We are primarily focused on safety, tolerability, and dystrophin. And as you would guess, when a company or when any drug development program is being started, you want to make sure that you have patient safety first and foremost in mind. And so we are not planning to add an arm right now. We're planning to do this relatively, modest size study in male participants to ensure that our drug is safe, tolerable, and is able to actually produce changes in dystrophin. And then from there, we have a much fuller clinical development plan that we are creating, in which we're going to have considerations like that, in terms of, what do we want to do more in the non-ambulant patient population, in the ambulant patient population, in other populations. So all of that thinking is ongoing right now, but we want to be very prudent about this first study to make sure that we can do what we think the drug should be doing.
Thank you. What is the frequency of dosing in this trial?
So it's every 4 weeks in every period of the study. So in the 24-week placebo portion, it's every 4 weeks. The open label extension after that, which is 24 weeks, and the 96-week long-term extension. So it is essentially monthly dosing.
Do you feel you'll focus on those who may need 2 drugs, such as those with deletion 46 to 52?
Sorry, I just want to make sure I understand the question. Focus on those who need two drugs. You mean 2 exon skipping drugs, or I'm not sure.
So yeah, I think that's the question.
So I think that I'm probably not the right expert to ask about double exon skipping. There's probably people that are I'm sure there's people more well-versed than me. It is a question that's come up. I don't know that the field is there yet in terms of how to skip 2 exons in a given individual, because there are certainly kinetics involved in that are probably beyond my understanding, to be able to do sort of double skip with 2 different exon skipping drugs. But we don't have plans to do that, and I think there's a lot more preclinical research that needs to be done in the field to be able to understand how to deliver 2 exon skipping drugs in a given individual.
Perfect. And then will you be working alongside third-party companies to capture real life benefits as opposed to just the data?
Absolutely, absolutely. And so that's, that I think has not actually been done enough in the field. There have been a couple of companies that have done a really, really nice job on capturing real world, data, like for example, PTC Therapeutics, in their STRIDE registry. So I think that we are absolutely planning to ensure that we are collecting real world data to be able to generate real world evidence to truly be able to characterize long-term, benefit, long-term value of this kind of chronic treatment. And so it'll also go beyond what we're doing now in terms of a select set of measures of strength and function, but we also want to characterize quality of life, other humanistic burden, resource burden. And so we absolutely are committed to capturing real world data. And frankly, it's not really optional. I mean, payers across the globe are going to require this. Some may require registries where you are looking at outcomes over a long period of time of 5 years, 6 years, 7 years. And so we are absolutely committed to capturing real world data. Sorry, Alexa, are you on mute?
Yeah, I am. Thanks, Ash. Those are all the questions that we have at this time. I'll give it like another 20 seconds while I close this up. But if you guys have any other questions, please don't hesitate to reach out to myself. My email is here on the screen, alexa@jettfoundation.org, and we will get those to Ash and Molly. And if they are able to, they will answer the question and we can get it back to you. Thank you all again for being here. Thank you, Ash. Thank you, Molly, for this presentation and helping us educate the community. And we hope to see you again on our other webinars. We have more coming up in March and later in the year. So thank you again, and we hope to see you soon.
Great. Thank you for having us.
Thank you.
Bye, guys.
Bye-bye. Take care.